All right, everybody. Here's where we talk about meiosis. All right, let's give this picture a superficial view for a second and take a look and see what's happening here. So clearly we have a beginning and we have an end. And you can kind of see what is happening as we progress along these blue arrows. So clearly from this first stage here, we have some change that's occurring. We can see that blue set of arrows forks into two different pathways a little bit later, about halfway through. And then at the very end, you can see two cells developing at the end of this line and two cells developing at that line. Now, that's a very superficial view, and we're going to get into what happens during every stage along the way during meiosis. Okay? But what stands out as happening overall is starting with one cell, eventually reaching a branch point where we have two cells going in the same direction and then finishing off with four total cells at the end. Let's get into how this works. First of all, meiosis is divided into two parts, meiosis 1 and meiosis 2. We're going to focus in on meiosis 1 right now, and that occurs in 4 phases. You're going to recognize these names because we saw them in mitosis a little while ago. Okay, so we have prophase, metaphase, anaphase, and telophase, all followed by the number 1. So this is round 1. Okay, now let's take a look and see what happens along the way in these 4 stages. Prophase 1 is a long one, typically occupies the most time, over 90% of all the steps during meiosis because of the multiple events that happen. Just like in mitosis, we start to see the chromosomes begin to condense. So we go from loose chromatin, DNA that we can't even see in the nucleus, to condensed, we call them squiggly jigglies, these things right here, structures which are wound up chromosomes, supercoiled DNA. Homologous chromosomes loosely pair up, aligned gene by gene. Now, in our prior talk, we talked about how, like in a karyotype, you saw 2 chromosomes, one from mom, one from dad, the homologous chromosomes lined up by size. Number 1 was the biggest, number 2, number 3. Well, they're going to find each other during prophase 1, which will take time, and they're going to line themselves up gene by gene. Okay? Now, when you do that, what you wind up with is essentially 4 sisters together called a tetrad. In mathematics and science, tetra means 4. Okay? So what we can expect then, and let me go ahead and I'll draw a blue and a red tetrad. Let me change the color real quick here to red. And whoops, here we go. They're going to line up, not out here in the cytoplasm, of course, but there wasn't room in there. So these 2 X's here are essentially a tetrad of chromosomes. You're going to have 1, 2, 3, and 4 in very close proximity, finding each other in the nucleus during prophase 1. Okay? And remember that these tetrads are also doubled up currently. So, you know, if I went ahead and made this a little bit thicker like that, that would be better representative to what we have here because they doubled up during interphase. Now, as you can see here, the picture shows we have the paternal chromosome in blue, that's the one from dad, and the red maternal chromosome from mom. And they're side by side with each other. We have big, medium, and small. And now that they've found each other, during late prophase, they're going to start heading towards the center of the cell. And remember, during mitosis, when these microtubules started connecting onto the chromosomes to help guide them to those central positions, we're going to see that again. As you can see, they make their way here to the middle. Okay, now another thing to notice is it's not as if all of mom's chromosomes wound up on top of the metaphase plate and dad's underneath it. It's completely random. So in this one, you know, mom's was on top, dad's was on the bottom. Same thing with this one. And then it flipped in this case. Remember, these are floating chromosomes. They're going to basically go where they want to go, and it's completely random in how they do that. Now, continuing on with prophase, that tetrad or group of 4 chromosomes, something very important is going to happen as they get together and then ultimately find their way to the middle and metaphase, and that's called crossing over. So in an earlier lecture, we talked about non-sister chromatids, which would be essentially this one here and this one here. They're not actually sisters with each other. They're sisters. This blue sister is sisters with this one. This red sister is sister with that one. And they're going to exchange. They're going to exchange DNA. Look at this. Their legs are going to cross over each other right here. And then it's going to snap. And when it snaps, look at what happened. We traded some DNA, right? Okay, so we have new genetic combinations from that crossing over event before they head to that metaphase plate. So where that happens right here is called the chiasmata. Okay, this X-like structure, that's the crossover spot. And other things that are occurring at the same time as this is just like in mitosis, we have the nuclear membrane breaking down. So here we can see it's intact in interphase. Here's early pro-metaphase, or we can say late prophase, and by the time you get to metaphase, it's not there anymore. Okay, so nuclear membrane goes away, nucleolus disappears, microtubules attach at the centromere region, which we had previously called the kinetochore, a set of proteins that sits outside the centromere. And here's a picture of that. Here's your supercoiled wound up DNA into chromosomes. Your centromere is the narrow portion in the middle, and then the kinetochore are these proteins outside. And these right here, these are your microtubules that are attaching it, and they're going to guide it to the middle of the cell. When they arrive there, that's metaphase 1. Those tetrads line up at the metaphase plate. Microtubules from one pole are attached to the kinetochore of one chromosome of each tetrad, and then microtubules from the other pole are connected to the kinetochore of the other chromosome. So you can see it on either side, here and here. Now, you can see them lined up, and again, there's no rhyme or reason for which one is on which side. You can see that these microtubules are now connecting onto the chromosomes in the middle. Collectively, it's all called the spindle apparatus, all of these including the centrosomes here. We have kinetochore microtubules are actually the ones that attach right here on the sides. And then remember, we also have these non-kinetochore microtubules, and we said these were the ones that stretched out the cell. So like if you have one coming from that side, one coming from that side, and right in the middle, they're pushing and pulling against each other, they're going to send the cell in opposite directions, elongating the cell. Okay, we've also dropped the term aster when we were talking about mitosis. This is here, these radiating explosion of microtubules out of the cytoskeleton, centromere. There we go. And after that pause in metaphase, what's going to happen is these homologous sisters are going to say goodbye to each other. Okay, now if you remember our talk in mitosis, we said they're going to walk up the microtubules with their little protein feet, or little motor protein feet, and as they do it, the microtubule disintegrates behind it, and they're going to walk their way back to each respective centrosome here. I put, why are there different colored tips on the chromosomes? Again, remember blue, here's red with a blue foot, here's blue with a red foot. Why does this happen again? Hopefully you remember that crossing over event. That's why these had those tips, because they exchanged with their non-sister homologs. Okay? And then in anaphase 2, as it comes to completion, we work ourselves over to telophase, and we'll recall some of those things as well. Okay, so in anaphase, pairs of homologous chromosomes separate. One chromosome moves towards each pole, guided by the spindle apparatus. Sister chromatids remain attached to the centromere and move as one unit towards the pole. There's a nice picture of it there. When they do reach those poles, we're now in that telophase stage of this division. And each half of the cell has now a haploid set of chromosomes, because one chromosome went to each side. But they're still made of two sister chromatids each. That's the difference, okay? The two chromosomal areas are pulling farther away from each other. Cytokinesis starts. We're well aware what that is at this point. That's the folding inward of the cell membrane, eventually to the middle where it separates the two cells, okay? All right, so now we got two cells that are haploid. Okay. A cleavage furrow still exists in these meiotically dividing cells as well. But here's a key thing that doesn't occur once we've divided the one cell into two, is that there's no more chromosome replication between the end of meiosis 1 and beginning of meiosis 2. That happened in interphase. Okay, we don't need to double up anymore, because what we're going to do is we're going to split what we currently have. So ultimately, these two cells here that are now the results of that process are going to split one more time immediately without replicating again. All right, and that brings us to meiosis 2. Again, 4 steps: PMAT, prophase, metaphase, anaphase, telophase, all with a 2 behind them now. Okay, so those two resulting cells right here, they're going to start at this point doing a lot of the same actions that we saw in mitosis. Well, like what? Well, they're basically going to gravitate and move towards the metaphase plate one last time. The spindle microtubules are going to help do that in both this cell and this cell. This is cell 1. This is cell 2 from our prior division a minute ago. There we go. Complete that too. Line up down the center one more time in metaphase 2. And you can see these are recombined chromosomes because of that crossing over event. Wind up going down the middle in both cells. In anaphase, remember what happened in mitosis. Sister chromatids split. Well, same thing's happening again. One more time, the chromosomes are going to walk their way to the centrosome region of each end of the cell. But look at the differences in the cell and the chromosomes that we have now. We got a red one with a blue foot. We got a pure red one. We got a blue one with 2 red feet. We got a blue one with a red foot. One with 2 blue feet. One with just 2 red feet of the medium size. So incredibly different combinations of chromosomes heading to each corner of what will become 4 cells in a minute. Okay, and then once those walk back to their respective ends, we then have cytokinesis one last time. The nuclear membrane will begin to reform, and we have 4 brand new haploid cells that are each genetically different from each other. Okay, if you look at the contents in each one of these cells, notice, let's look at the big chromosome. Here's one big blue in this cell. Here's a big blue with a red tip. Here's a completely red big chromosome, and here's a red one with a blue tip. That's 4 different cells just based on that large chromosome itself. So as a final note, mitosis 2 is very similar to mitosis. All the same actions occur there. All right, let's go through the little tiny points in each one. Prophase 2, another spindle apparatus forms. Chromosomes condense after a brief unraveling at the end of telophase 1. In the late part of this stage, the chromosomes will move towards that metaphase plate. In metaphase, we know what happens. They line down the center there. And because of crossing over, the 2 sister chromatids are no longer genetically identical. Okay, we got a whole bunch of different recombined chromosomes now. The kinetochores of the sister chromatids attach to microtubules extending from opposite poles on the left and the right. In anaphase, those sister chromatids that are now down the middle are going to separate, and each one is headed towards their respective poles. Telophase 2 in cytokinesis, everybody arrives at their end of the cell. Nuclei begin to reform, so that means the nuclear membrane comes back. Chromosomes begin decondensing, so you don't have to keep them as these wound up supercoiled chromosomes anymore. You can unwind them, let them relax a little bit as unwound DNA. And then the cytokinesis breaks those 2 cells into a total of 4. Okay, so again, if we're just looking at one chromosome in a cell and it goes through these divisions in meiosis 2, even with one single chromosome, you can see there's 4 different possible outcomes. You have the all red, the all blue, the red with the blue foot, and the blue with the red foot. We'd call these 2 in the middle recombinant chromosomes, and then these 2 on the ends would be non-recombinant because they weren't affected by it. But what is the bottom line here is you got 4 genetically unique cells that are different from the parent cell. All right, so if we compare mitosis and meiosis, meiosis will conserve the number of chromosome sets, producing cells that are genetically identical to the parent cell. We made clones, we talked about. Meiosis reduces the number of chromosome sets from 2 to 1. Okay, and it will produce cells in the end that are genetically different from each other and the parent. And another thing we could say about meiosis is, remember, these happen in the gonads. Okay, gonads are the sex organs, right? And that would be the ovaries in females, and it would be the testes in males. That's where this process happens, meiosis. But mitosis is everywhere else, okay? Happens right everywhere but the gonads, okay? So most of your cells use mitosis and not meiosis. And the last thing, the mechanism for separating sister chromatids is virtually identical, meiosis 2 and mitosis. So we could, put one against the other and look at some of the things that happen here. DNA replication happens in both during interphase before they start. Number of divisions, mitosis has 1 and meiosis has 2. So it's good to do a compare and contrast here. They're the same because they each go through PMAT, right? But meiosis goes through it twice. Synapsis of homologous chromosomes. Synapsis was where those tetrads got together, and then the non-sister chromatids crossed over in the middle. Okay, so crossing over was the big part of that, of synapsis. Number of daughter cells, mitosis gives you 2, genetically identical cells, clone cells, and meiosis gives you 4, and each one genetically different from the parent and each other. Their roles in the body, growth, repair, asexual reproduction with some particular cells, we said that bacteria do binary fission, but some single-celled organisms do go through mitosis to produce a whole new individual. And in meiosis, we're producing gametes. We're producing sperm and eggs. And we're reducing the number of chromosomes by half and introducing genetic variability. Three events are going to be very unique to meiosis, and all three happen during meiosis 1. So synapsis, where they line up with each other, and then the crossing over. They physically connect, exchange genetic information. At the metaphase plate, instead of there being chromosomes just made of 2 sisters, we have homologous chromosomes lining up, what we call homologs for short, instead of those individual replicated chromosomes. In anaphase 1, it's those homologous chromosomes instead of sisters that separate. Okay, so they pull away from each other. And we're just going to leave it there for meiosis. Hopefully that makes sense. Obviously, read as much as you can on your end to back this up. Clearly, you probably wanted to pause this a whole bunch of times to take it all in, but ultimately the process is again cell division, but cell division that produces unique variability in offspring at a later time. Okay, all right. So that's your talk about meiosis, and I'll see you guys at the next one.